Welcome back to Quick Look on Solid Phase Extraction. I'm Paula Orens. In part 2, we continue our discussion on SPE and the long-standing dispute of vacuum or positive pressure. With me, I have our 3 SPE experts who will continue to help us answer that question. Welcome back, Melvin Blaze.
Hi.
Ken Berthollet, both from our chemistry R&D organization, and Katie Brennan from Water Scientific Operations. In our last discussion, we spoke about some basic SPE processing woes and why it's important to overcome them, the most important of which was flow rate. Kenny, do you find that positive pressure or even vacuum have any benefits when it comes to controlling the flow rate?
Yeah, so in the work that I've done, I found that positive pressure is much better at controlling the flow rate. Because I can set at least the positive pressure manifold that Waters offers, it can be set to very low pressures. It allows me to better control how fast my sample is going through my device. With a vacuum manifold, you're kind of stuck with however strong your pump is. And then there's a lot of extra accessories involved with a vacuum manifold, so it's not as easily controllable from my experience.
Melvin, have you found any other benefits from using positive pressure?
Yes, I totally agree with that, with whatever Kenny mentioned. So especially with plate formats, I think positive pressure is very, very useful in fine controlling the flow that you want for your load step as well as for your elute step. Whereas in the vacuum manifold, I also see that in terms of cartridges, there is an issue with cross-contamination that's quite often seen because you connect the same cartridge to the same stopcock valve that you have on the manifold. So when you move from one experiment to the other experiment, the stopcock valve is still the same. So you need to clean up those valves very well before you move to the next experiment, which you don't have that issue with positive pressure manifolds because everything is a disposable consumables that you use for positive pressure manifolds. So I think in that way, if you see overall, I prefer positive pressure manifold versus vacuum.
That's a really good point, Melvin. I hadn't considered the potential contamination issue of the different engagement between the cartridge and the manifold itself.
Yeah.
So while we're on the topic of vacuum manifolds, we talked a little bit about the downsides, but are there any benefits to vacuum manifolds over positive pressure, or have we found our answer?
Depending on the application, positive pressure isn't always applicable. So if you're doing, water analyses where you have, a liter or 2 of water that you need to push through a device to concentrate your analytes, positive pressure just isn't that compatible just because you don't have access to the top of your cartridge. Vacuum manifolds have the top of the cartridges still accessible. So for that, at least that application, vacuum manifold is still the preferred method, I believe.
Larger volume samples.
Yeah.
Okay. Katie, what about you? Do you find any benefits of one over the other?
I would say the same with Kenny, with those larger volumes and with those larger cartridges that you would want to use with a large vacuum manifold. You can't necessarily use that with what's on the market right now for positive pressure.
Well, I'd like to thank you for joining in today's Quick Look on Solid Phase Extraction. Be sure to look out for the third and final part of this series. We came close today, but in the third part, we will conclude the discussion and find the answer to the question, vacuum or positive pressure. For more information about different tools for processing SPE successfully, please visit waters.com/auto. See you soon.